Hi, this is Mr. Warchke again, and today we're going to start with the second genetics screencast. I know you're excited. In the second genetics screencast, we're going to start off with Mendel's second law, which is the law of independent assortment. Now, this deals with the chromosomes being shuffled, and they move independently of each other. So in the formation of gametes, each gene or factor can move independently of other genes or factors. So you have A alleles, they move independently of B alleles, which move independently of C alleles, which move independently of D alleles. Each chromosome, which contains the genes, will move independently from the other chromosomes. So this allows the chromosomes to shuffle each other and gives us an unbelievable amount of variation in the population. But we digress. So we're looking at more than one trait in each cross. So instead of just saying A's, which is really easy, now we're including A's and B's. So different traits, maybe seed color and seed form. So we're going to take a look at round, which is seed form, and wrinkled peas, and yellow and green peas, which are color. So we're going to say that a truly homozygous round and green pea is crossed with a homozygous wrinkled and yellow pea. So in the F1 generation, all you can get is one big A, one little a, one big B, one little b. So every pea in the F1 generation will be round and yellow because they are heterozygous. Dihybrid, two heterozygous. The gametes, you have to figure out the gametes because that's probably the most important step in figuring out your Punnett square. Remember, so you can only get one A, one trait for seed form, and one trait for seed color from each parent. So when they combine, you'll get two genes for seed form and two genes for seed color. So what are the different possible gametes that you can have? Well, this parent here can give a big A or big B. That's one possibility. Or a big A and a little b, okay. Or this parent can give a little a and a big B, or a little a and a little b. So those are the gametes, the sperm or the egg. And each parent, since they have the same genotype, same collection of genes, they will give the same identical gametes. So here's the Punnett square. We have four gametes on the top, four gametes coming down, and we combine these gametes with these gametes to get that child, that possible child. We combine this gametes here with this gametes here to get that possible child. Now remember, if you're dominant here, you're round. If you're dominant here, you're yellow. So these are the genotypes and phenotypes of each of the possible offspring. And you're definitely going to have to get this down. So take some time, hit pause, and have fun doing a big Punnett square. And if you think this is big, ha ha, I laugh at you. So I'm going to give you Warchke's method of gamete determination to help you figure out the gametes, because once the gametes are figured out, Punnett squares are really easy, or as we used to say up in New England, wickedly easy. So all you need to do to determine gametes is my method. If you can set up a square, and you can do my method, and you can combine things, you're good to go. So my method is equal to, so the total number of gametes, the total number of different gametes is 2 to the n, which the n equals the number of heterozygotes. So I'm going to do big A, big B, little b, big D, little d, and big E. Why not C's? Because capital C's and small c's are really hard to see. So we're going to do that. So there are 4 different heterozygotes. There's one heterozygote here, 2, 3, 4. See that? 4 heterozygotes. So it's 2 to the 4th. That is equal to not 8, but 16. So 2 times 2 is 4, times 2 is 8, times 2 is 16. If you did 2 times 4 and you got 8, you're wrong. It's 2 to the 4th, which is 16. So you have 16 total gametes. Now you know that 8 of them will have a big A and 8 of them will have a little a. You know that 8 of them will have a big B and 8 will have a little b. Same thing with the D's. 8 will have big D's, and 8 will have little D's, and 8 will have big E's, and 8 will have little E's. So you need a way, a method to write these letters down so you get all the possible combinations. All you need is a pattern. And I'm going to give you that pattern. So you take the total number of gametes, which is 16, and divide it by 2, and that's for the first letter or the first gene. So you're going to write down 8 big A's and 8 little A's. Right in a row. 8 big A's, write them down. 8 big A's, 8 little A's. 2 different columns. Now, you're not going to write down 8 big B's and 8 little B's. No, because that's not right. So you have to change the pattern a little bit. So what you're going to do is take the first letter number, which is 8, divided by 2, which is 4, and you're going to write 4 big B's, 4 little B's, 4 big B's, 4 little B's. Right next to the A's. So when you have 8 big A's there, you can write 4 big B's, and you can switch and write 4 little B's. All right. So then when you get to the little A's, you can write 4 big B's and 4 little B's. Do it. Now for the third letter, you're going to take the second letter number divided by 2. So 4 divided by 2 is, yes, 2. Very good. So you're going to write 2 big D's, 2 little D's, 2 big D's, 2 little D's, 2 big D's, 2 little D's. And finally, the last letter, you're going to take the third letter divided by 2. 2 divided by 2 is 1. So you're going to alternate E's all the way down. Big E, little E, big E, little E, big E, and all the way down to the end. You're going to get something that looks like this. Right? You have 8 big A's, 8 little A's, 4 big B's, 4 little B's, 4 big B's, 4 little B's, 2 big D's, 2 little D's, 2 big D's, 2 little D's, 2 big D's, 2 little D's, 2 big D's, 2 little D's. And I'm not going to do E, E, E. So alternate the E's all the way down. And if you take a look at that, you will notice that every single gamete is different. You have come up with all the different gametes. And this works 100% of the time as long as you're writing down heterozygous. You're thinking, well, what happens if I had a homozygous F? Well, then every one, big F. If you have homozygous big F, then every one of these things is going to get that big F. It doesn't matter. Think about it. It's beautiful it's in its simplicity, and no one else knows about it but my students. So test cross. In a test cross, you're going to determine if a dominant individual is homozygous or heterozygous. How do you do that? By crossing it with a recessive individual. If they're homozygous dominant, cross with the recessive, all the offspring will be dominant because all the offspring will be heterozygous. If the individual is heterozygous and if you cross it with a homozygous recessive, then 50% of the children should be dominant and 50% of the children should be recessive. That's how you know if the adult is dominant, or homozygous dominant, or heterozygous dominant. So that was basically Mendel, and that was earth-shattering. And we could talk about Mendel and Darwin just missing each other, and that we could talk about that during evolution. Remind me later. But we're going to talk about modern Mendelian genetics. So first, why are recessive genes recessive? Well, recessive genes are mutants of the dominant gene, and they produce a non-functional protein. That's why recessives are recessive, because the protein that they produce isn't the right shape. It doesn't have the right function. Lethal recessives. So in a lethal recessive, the individual cannot survive. So the protein is necessary for survival, and if they have 2 copies of a non-functional protein, they will die. Examples of Tay-Sachs, when they have a fatty buildup in the brain, and kids die by the time they're 4 years old. Or cystic fibrosis, where individuals used to die when they were in their mid-20s because their mucus was thick and sticky because they'd had an improper ion transport protein. The third example of modern Mendelian genetics is partial or incomplete dominance, and when the heterozygote is a mixture of the 2 homozygotes. A heterozygote is a mixture of the 2 homozygotes. So you can have a red flower here and a red flower here, and the heterozygote will be a mixture of red, and you end up with pink. Yes, like in snapdragons. These aren't snapdragons, so you pinch a snapdragon, it looks like it's opening its mouth. Womp, womp, womp. Ah, there's a snapdragon right there. You pinch the side of the snapdragon, pinch it right here and on the other side, and it unfolds. So right, we have red, white, we end up with pink. Look at that. P generation, F1 generation. Almost like I know what I'm talking about. Red gives a big R, white gives a little r, big R, little r, heterozygote. Codominance. And hopefully we've talked about this when we did that law of segregation assignment. So codominance, there are two alleles are expressed equally, and that's blood type. So there are three alleles in blood types, A, B, and O. Three genes for blood types. A codes for the A protein, B codes for the B protein, O codes for nothing. O is recessive to both A and B. So if you have A blood, you can have AA or AO. B blood, BB or BO. Now, if you have A gene and B gene together, it's called AB blood because both the A and the B are expressed equally. That is codominance. Codominance. The only way to get O blood is if you have two small O's or two O's. Multiple alleles. So we've talked about seed form. Well, Mendel only had two alleles for each trait, right? Big A, little a, big B, little b. Brown, wrinkled, yellow, green. But blood type, there are more than two alleles. There are A, B, and O. Multiple alleles. Epistasis. Now, this is a crazy one in which one gene controls the expression of another. For example, coat color in mice. So we have the big B is black, little b is brown, but there is another gene at another location that codes for pigment. If the mouse has the black genes, if it has no pigment, it will not be black, it will be white. It'll be an albino. Same thing with brown. If it's brown, if it has no pigment, that doesn't matter. It will still be white. So if we have two black mice that are heterozygous for the pigment gene as well as the coat color gene, what is the phenotypic ratio? Well, you have three colors. You have black, brown, and white. So we got 1 black, 2 black, 3 black, 4 black, 5 black, 6 black, 7 black, 8 black, 9 black. 9 black mice. We have 1 brown. We have to have 2 browns, 3 browns, and then we have 4 white because they have no pigment. So it's 9 to 3 to 4. 9 black, 3 brown, 4 white. Doesn't seem like the easy ratios that we've had before. Pleiotropy is just like epistasis, except one gene controls the expression of more than one trait. So for example, the coat color in cats, a white cat will not only have blue eyes, but they will also be deaf. Yes. And nature versus nurture. So when the environment controls the expression of traits, these are called multifactorial. For example, spina bifida and cleft lip and palate will only be present if certain environmental conditions are met. And we don't want to meet those environmental conditions, so we do everything to control our environment so that doesn't happen. For example, spina bifida, women take folic acid when they're pregnant. Now, this deals with epigenetics, which we will talk about later. Now, epigenetics is how the environment affects the expression of genes in people, which is why identical twins, after a while, aren't identical. But we'll end there for today's screencast. And I know you're excited for the next one. Have a good night.